This is the reason we're here today, is to thank you, to give an accounting to you of what we've done with the study and a report of what we've done and what we plan to do. So with that, let me turn it over to turn it over to Dr. Nate. Thanks, Rachel. All right, Dr. Nate Chin, go for it.
Thank you. Can everyone hear me okay? Okay, wonderful. And thank you all for being here. I have the privilege of being able to talk about updates in Alzheimer's disease in 2023. And there are a lot has happened in the past few years. And this field has been around for quite a long time. And there have been some successes and many that are not. But I would say in the past 10 years alone, the field has grown at such a rapid pace it sometimes feels overwhelming in trying to keep up. And so I'm going to summarize some of these key things today. And really I want to start with the fact that Alzheimer's is quite present in our communities, in our country, and in the world. And when you look at just the United States, roughly 6.5 million people are affected by Alzheimer's disease or a related condition that affects the brain. And that is a rough estimate. If you look at a different source, it might be 6.7 or closer to 7 million. But these are individuals who have dementia that's related or caused by Alzheimer's changes. If we start expanding that definition, if we look at other causes, the number gets higher. If we start looking at a condition called mild cognitive impairment, that number goes to 14 million. And if we start thinking about people without symptoms, but they have the biological changes in the brain, and now that number looks closer to 40 million. And these are the things that we're able to say more confidently than we were before because of these biomarkers that Dr. Johnson has spoken about already. The ability to detect change in the living brain even before symptoms. And that's really what RAP has helped push forward in the field, and that we'll explain in a few minutes. So as we define Alzheimer's disease, we're looking at it as two proteins that accumulate in the brain. One is amyloid, it forms outside of the brain cell, and the second is tau, or neurofibrillary tangles, and that forms inside of the brain cell. And those two proteins are how we define Alzheimer's disease, without having symptoms, just the presence of those two at abnormal levels. On top of that, we can then start explaining the more important aspect of the human experience, the symptoms that someone may develop, and those are called clinical stages or syndromes. But because we're able to look at these proteins, there is this long pre-symptomatic or pre-clinical stage. And RAP truly is at the forefront of this. RAP has truly defines this and continues to fine-tune what we see and what the world has been looking at. And we can do this with the things that you all have contributed. Those lumbar punctures, so you can look at the spinal fluid in the brain that bathes the brain. We can look at those proteins. For those that have been willing to go into a PET scan, it's a fancy imaging technique. We're able to identify those proteins through an amyloid PET scan or a tau PET scan. And now we're getting very close, or already there, for blood. A simple blood test can do the same thing, and possibly more. We still have to take into context lived experience, and that other things are happening to our brains, not just Alzheimer's disease. So there are other conditions of the brain and below the brain that can affect our thinking, and we need to differentiate that. We need to identify that. And so RAP is expanding and looking at these factors too. And we also know that when they occur with Alzheimer's disease, a person's experience, those symptoms, the course that they have is going to be different, and oftentimes can be faster. And then the last thing, which I'm sure many of you are already aware of because it's very popular in the news, in the media, is that we have new therapies, new medications that have gone through clinical trials that are now partially FDA approved and are frankly talked about all the time. And there's a new one that just came out this past week. So what I mean by new therapies? Well, there are two that are FDA approved right now. We anticipate 3 very soon. But the first one is aducanumab. That created quite a lot of publicity because there was a lot of controversy with it. It still did what it was supposed to in many of the participants, which is remove that first protein of amyloid. The second drug is lecanemab, and that one people are not really debating. It can effectively remove that protein, and I'll show you a couple slides in a second. But how it works is the mab on the end of lecanemab or aducanumab stands for monoclonal antibody. This is not new. It's not a new technique. Rheumatology, oncology, many fields, allergy, are using these types of therapies. But we can create this medication, infuse it into the body, and very much like your own immune system, these are proteins that attach to specific parts of a target. And in this regard, it's an amyloid protein and a certain target, not the whole protein. And so that's why there's many of them, because they can target different things. They attach to us, they go into the brain, they then stimulate your own immune system to say this is a problem, come. Your immune system comes, breaks down this protein, clears it out of the body. Which is pretty remarkable. But I do want you to think of it like chemotherapy. It's an infusion. So there are some limitations to this. There are certainly side effects that we have to be very thoughtful of. But what does the actual evidence show? And so in this figure here, I want you to pay attention to the horizontal line representing time. So 0, 3 months, 6 months, 12 months, 18 months. 18 months tends to be the standard duration of these clinical trials. 18 months on a therapy or on what we call placebo, not the medication. The black line is those on the placebo, so they're not receiving therapy. And the green line is the individuals on the lecanemab. And the horizontal or the vertical axis is the amount of amyloid that is removed. So it looks like it's starting at 0. These individuals actually have about 75 units on a scale of 0 to 100, so it's quite a bit of amyloid. And so you see that that black line continues to slowly go up. So those individuals, and that makes sense, they're not on the therapy, so the amyloid is slowly accumulating. But the green line, well, that represents lecanemab therapy. And you can see already at 3 months, there is a separation, statistically significant. The drug is removing amyloid based on these tests. And by 18 months, we certainly see a pretty significant separation. And they remove enough, about 60 units of this amyloid, so going from, let's say, 75 down to 15. Well, 15 is undetectable. So for 2/3 of people on lecanemab, this drug went from having, took enough amyloid out of an individual that they are no longer detectable. So it was very successful. And this is what the FDA based their approval on. Not on the lived experience or symptoms, but based on this fact alone that we can do what this drug is supposed to do. But it's more than that. And so we need to know what people are experiencing, right? Because the ultimate outcome is how are people living with this condition? How can we make this a chronic condition? How do we stop it from progressing? How do I ideally bring back a person's abilities? And so in this regard, the same time frame on the horizontal line, so months, 3, 6, 9, 12, 18, 15 even, and then the black line representing people with the placebo, the green line representing people on the therapy. In this regard, that vertical line is not the amount of amyloid, it is a person's thinking and functional symptoms. And this is actually the same questionnaires that you as a participant or your family and friend as the study participant that you're filling out for us. These are standardized, validated, important questionnaires so that we know what a person's experiencing. And as you see, the people on the placebo, the control, they're declining, and they're declining at a pretty consistent, steady rate. And the people who are receiving the therapy, the green line, well, they're declining at a slower rate, or a change. It's not as much of a decline. And you can see the circle, so even at 6 months we can see this, and certainly at 18 months we can see this. And so they're still declining, so this is not a cure. It does not stop the disease from progressing, but it certainly is changing what that course looks like. And at 18 months, there's a 27% reduced decline in those who are on the therapy. 27%. And so what that means, if you ask one of our statisticians, or certainly one in the clinical trial, it's roughly 6 to 8 months of keeping someone within a stage, mild cognitive impairment or mild stage dementia. And for some, that's going to be quite significant, quite meaningful. But that is what the field is grappling with. What is clinically meaningful? and it's going to be debated going forward. Now from this, we now, as of this past week, have a third drug, not FDA approved, but called donanemab. It's a mAb, so it's still that monoclonal. And what they did at the end of 18 months is they showed 35% reduced decline. Now they used a slightly different scale than what you all have filled out in your visits. But if we then look at those exact same scales, it was 36% of a decline. So they're saying their drug is more effective. And so this era of competition, this is a good thing. So we're glad to see that a different medication is showing more efficacy or more of our outcome that we're looking for. They did something very interesting in those that stopped, those that went undetectable at 12 months, they then stopped giving them the medication. They still followed them to the 18th, and there's still ongoing improvements or slowed change 6 months later, even without getting them the therapy. So they're really challenging the total duration, which is great because less time on therapy is going to be a good thing for us. One thing that was very interesting, I know Dr. Johnson's like, move on. One thing that's super important. One thing that's super interesting about this is how they got into the study. Well, we need to know that they had amyloid, but they also measured their tau level, that second protein. And my understanding is that that hasn't been done before. Is that right?
That's right.
Okay, so that hasn't been, and that they're stratifying people based on the second protein. So yes, you had to be in because of the first one, but then where you were in the study was based on the second one. And certainly when you have a little bit of tau, they found more benefit with this medication. So we're getting closer and closer to precision medicine. But how do we even get to this point where we can start talking about this? Because the participants willing to come in year after year to do PET scans, to do lumbar punctures, so that we can make sure that these tests are valid, that they're reproducible, that they matter to people. So we have to know that the proteins are there and that these drugs can do what we think they can do. And so we're continuing to do that at RAP. We have to measure these proteins. We have to do those lumbar punctures and those PET scans. So as much as I'm excited about blood, if one of the things you're thinking about is maybe I don't have to do that anymore, the answer is no. We still need you to do that. We need to know that the blood eventually is as good as, or as good as the lumbar punctures and PET scans. We still need these things. And this is what's being used in clinical trials right now. And so those are really important. These are still very good ways of measuring it. But blood is certainly something we're very excited about. It will accelerate the field. It will make clinical trials better. We also still need to do all the questionnaires and the cognitive testing. And I know people don't like the cognitive testing, but what RAP is, I appreciate the feedback. And I know they're long, and I know they're difficult, and they're meant to be difficult, but RAP is famous for being a cognitively driven study, meaning our outcome is you and your thinking ability and trying to preserve, improve, maintain that ability. And so other studies use much shorter ones, and it's not as good. And that's why people from across the world contact Dr. Johnson and the team to say, can we look at your data? Can we do this? Because we know that your results are valid, and we know that you are rigorous in understanding thinking ability. So we still have to do that. I don't imagine that will ever change. Sorry, that's a down for you. But that is really what matters, right? It's our lived experience, our thinking ability. So we'll continue to do that as well as our physical exams, and I'll explain the meaning of that as well.
He's got such a great way of delivering bad news, doesn't he?
Here's some good news, here's some bad news. Let's go back to some good news.
Right.
So, how we think about Alzheimer's disease is really based on studies like RAP and other of our colleagues' studies, looking at people over time. And we are now able to say with more confidence, this is how we view the progression of Alzheimer's disease. It starts with amyloid, so you see a picture of an amyloid PET scan. Dr. Johnson will show you more. After some point in time, which RAP is really working on, then the development of tau protein. After that, sometimes people are going to have brain cells that are going to die at a faster rate. That's neurodegeneration, brain cell death. Those three things are about the brain mind, because not everyone's going to have symptoms with this. In fact, most people won't have symptoms yet. This is preclinical. This is that pre-symptomatic window. that Dr. Johnson's referring to. And it's very rare. For some people it's 10 years, but for others it's 30 years. So it's a huge opportunity for us to not only understand what's happening, but then to intervene. At some point, at least in the current state, people are going to progress, or some people will progress past that green line. And that's sort of our clinical threshold. And then perhaps they see me in the clinic and they're diagnosed with mild cognitive impairment, and they progress and they're diagnosed with dementia. But this is how we see progression, but this is how we overlap biology with the lived experience of a human person. So what has RAP done to contribute to this huge advancement in our understanding of Alzheimer's disease? Well, first, let's recognize that RAP is the largest and longest standing running study of individuals at risk of Alzheimer's disease. So that's you. So thank you all for doing this. Decades. How many people in these studies where they say decades of dedicated participants who come in every other year to do the things that we do? Lumbar punctures, testing, blood tests, exams. Do long visits, so thank you all for that. But that is what RAP is known for. Dedicated, loyal participants who've been willing to give so much of themselves and their time, so that very rigorous, very particular scientists like Dr. Johnson can look at the data and try to understand what's happening to people. The other thing about being a longitudinal study is that RAP is also a registry, meaning that other scientists from across the campus, across the country, across the world, can then reach out to RAP, to Dr. Johnson, and ask, well, can I look at this data because of this particular scientific question? Could I potentially engage with these participants for this reason? And it's not a everyone can do this. It's a very rigorous process of making sure that those studies are valid. Because nothing with RAP can be not valid or not legitimate or not rigorous. So RAP participants are from all over. I mean, many of them probably started in Wisconsin, but now have dispersed. And so we see that people from across the country are still part of RAP. Some will fly directly to Wisconsin just to do the RAP visit. It's pretty remarkable. We would like to have better access, so we'd like to get to RAP participants at some point in the future. And you'll see there's someone there from Hawaii. I'm happy to visit that Dr. Johnson will pay for me to fly to Maui so that I can conduct that visit. Take a team of us and some of the staff in the back that can take me. We're trying to improve access. We really want that continuity. And so we will continue to do that and work on ways to do that. But you can see, as far as our RAP statistics go, I mean, RAP is an incredible group. Average age of 67. So young. There's a higher percent. Remember, I'm a geriatrician, so I see people in their hundreds too. So 67 is very young. There's a good percentage of people, I'm just going to stand over here. There's a good percentage that are women that have a family history. We are improving our underrepresented population to improve representation and diversity, so 18%. But you'll see in the middle row, RAP people have chronic diseases like everybody else. Blood pressure, cholesterol, obesity, blood sugar. Now the bottom row, those are some of those biomarkers. So ApoE4, that's that genetic risk factor. So 39, 40%, that's higher. That's intentional with the family history. So we're recruiting individuals that are going to have more of that. But only 26% have elevated amyloid in their brain. And when we look at our sister studies across the country, 20 to 30% is what we anticipate for people. And only 12% have tau. So it's still a smaller number. And I think most importantly, only 6% have thinking change. So RAP participants are quite healthy, which is great. And we need to understand why that is. Perhaps there's something that RAP is doing, RAP participants are doing, that others in the world need to know. I think the big number here is the one we darkened in the second row, 12. 12 years as the average length of a participant. 12 years of continuity of repeated measurements and data, including biomarkers. And it's because of that that we can talk about progression. It's because of that we can talk about some of the fancy things that Dr. Johnson will speak to next. So one of the other things that RAP participants have contributed, lots and lots of MRI scans, 1,900, over 1,000 amyloid PET scans, 680 tau PET scans, almost 7,000 research visits. All those visits have hundreds of data points. You can imagine the systems that are needed for that. 750 lumbar punctures, 6,500 blood samples, 117 people, scientists, have requested data from across the world, leading to over 200 publications. I will say here at this point, the data that is shared is only the data that you have given permission to share. It is very much protected in the UW system by our institutional review board, and we make sure that if you've given us permission, we make sure the science is valid, and then it's shared with those individuals. The last thing is nearly 600 people have enrolled in the brain donor program. And as great as biomarkers are, the brain, the autopsy, that is our definitive gold standard. That will never change. We will always need to understand the other things that might be happening in the brain. And we have the brain donor table back there, so if anyone has any questions, Jay can raise your hand. Jay is there to answer any of those. It's still a critical piece to this. And so with that, I'm going to turn it over to Dr. Johnson.
Thank you, Dr. Chin. It's really incredible to see that, to see all those numbers and the way the study has accumulated over the years. And as Dr. Chin said, it's decades worth. Started with Dr. Mark Sager in 2001, and he retired in 2014, and I took over at that point, and will someday pass this on to somebody else, I hope. But this is incredible, and we approach this with a sense of mission and purpose, about discovery, about understanding the biology of this disease, but also understanding you and treating you with respect, the way you want to be respected and engaged with. So we want to listen to you. We're always open to your feedback. We want to announce to you the exciting news that we are funded for another 5 years by the NIH. And this is really a fantastic development. We'll be telling you about some of the changes. There's a few changes that we want to introduce as this field has progressed. We will stay up with that and try to stay ahead of it a little bit as we see the trends from all of you. Well, let's talk about some of the discoveries that we've made and some of the questions that are on our mind, and some of the data that we have generated. We've discovered new ways and more technical ways of understanding subtle cognitive change. Before this category of mild cognitive impairment, we can see change, and that's because of the longitudinal part of the study. We've discovered a way to identify when this disease starts. When does amyloid begin? It's going to be different for everybody, and I'll show you that in a second. We've helped in the discovery of genetics. Genetics is a field of study that takes lots and lots of participants. So lots of cohorts like RAP and many others across the world have been part of these genetic studies, and they've revealed new genes and new molecules involved in this disease and other diseases. And with all of this, and anchoring it to when Alzheimer's begins, we can now study in a more realistic way. We can study resilience, study susceptibility. We can understand why some people continue to have normal lives in the face of having amyloid in their brain. And that's something we really want to know more about. And that's part of our agenda of burning questions over the next few years. We want to make sure that our discoveries are generalizable to all communities. That's a paramount scientific mission for us, an imperative as we do our science. We want to make sure it's generalizable. And we want to understand how lifestyle and health determinants and social determinants, our environments and our neighborhoods that we live in, affect our brain health and affect our cognitive functioning and affect the AD proteins that may or may not be happening. And as Dr. Chin mentioned in the beginning, we want to understand other diseases that could be co-occurring. And as the technology, as the biomarkers get better at these other diseases, we will be implementing those so that we can tease it apart. We know that when we look at brains under a microscope, and this is why the brain donor program is so important, that people who have definite Alzheimer's in their brain, about 60% of the time, they have at least one other protein abnormality in their brain That could be Lewy body disease. It could be other kinds of dementias that are in the brain. And we won't know that unless we get the brain at autopsy. And we are hopefully developing these tools to study that in life with the biomarkers. And all of this will lead to a more personalized approach to healthcare. And by being in this study, by being the pioneers that you are, you're helping, literally you're helping your neighbors, you're helping your families, your all the people who will be coming after you are going to benefit from your generosity and the way you volunteer for this kind of study. Well, let's talk about what shapes the preclinical window. We'll talk about blood markers. We'll talk about tau proteins, and so let's dig into it a little bit here. Let's start with these PET scans, and some of you have done PET scans. Can you raise your hand if you've done any of these PET scans? Incredible, thank you for This is the value of these scans. So we have two people here, one on the left, one on the right, and we have an amyloid scan and a tau scan. On the top is the amyloid scan, on the bottom is the tau scan. And you can see that the one on the left is full of red colors and orange colors. These are where the scan is detecting these proteins, the amyloid protein and the tau protein. The person on the right, even though they have memory symptoms, they don't have any of these proteins in their brain. And this, even at the back of the room, I think you can appreciate the clarity that this kind of method can provide. And let me show you some examples of how we can use this. So we can categorize you who've done these PET scans in terms of, is your amyloid elevated or not? Is your tau elevated or not? And that gives us 4 groups, roughly, of people who aren't elevated at all. And that, if you look at the 4 boxes on the left, that's the gray group. They don't have either of those proteins. And what we're seeing there is cognitive trends over time, where time is represented by age. And the y-axis on those, the vertical axis, is cognition from the RAP measurements that you've done. We don't see much of a trend in that gray box in the bottom left. And that's because they don't have either of those proteins. Anything that we see there would be attributable to normal aging or to other diseases that we're not able to measure yet. And that's why it's kind of a cloudy looking box. And the 2 diagonals, the blue and the green boxes, those are people who have one of the proteins in their brain but not the other. And we don't see much of a trend there either. But the red box, where we see these are people who have both proteins in their brain. And again, each of these lines represents change over time in a person. Each line is a person. We're seeing a fairly clear trend on the person level, such that cognitive change in these people who have both proteins happening in their brain, cognitive change is happening quite early in our 50s and 60s. And it continues to affect our cognitive health. And so these person level data points are, that's borne out by the statistical graph on the right. The group in red that have both those proteins in their brain, they are declining 3 times faster than the other groups. So that is that's part of the value, and that's a clear demonstration of why these PET scans are so important. They help us understand not just the diagnostic picture when you come to the doctor's office, but what might be happening, what may have happened in the prior decade in a person's lived experience. Well, with all of these PET scans that we've done, we've really made a major discovery. 500 people in RAP have done these amyloid PET scans, and that number grows every day, thanks to our PET imaging team who's here at the back of the room. And if you haven't had a PET scan yet, and you'd like to get involved, please talk to our team in the back. This has resulted in a major finding, and this is just an example of this. What we see here on the left in these line plots, this is now amyloid accumulation in people who've had PET scans, and each line represents a person who's had more than one, and we connect these lines together to see these trends. And what we see in red are the people who are amyloid positive or elevated. And their trajectories over time are pretty predictable. What differs really is the heterogeneity and when that happens on the x-axis, which is age. Some people get this disease, or the amyloid proteins, early, in their 40s and 50s, and some people it takes much longer. But this happens in such a predictable way that we can fit a mathematical function to this, and that's what you see in that plot on the right, such that we can put everybody on the same curve. And that means in the future we can do a single scan and actually fit somebody to this curve based on a single PET scan, get an idea from that single PET scan how long a person has had this disease and the age at which this disease began for that particular person. And that is a discovery that we've made with RAP, and that's thanks to all of you. But there's something I want to point out, and that is that all of science requires replication. We can't just make inference about a single study. There could have been some bias, there could have been some fluke in that study, so we need to look for replication. And I want you to take a picture of this in your mind. This is us. And now let's look at our competitors or collaborators down at St. Louis, at WashU University in St. Louis. This is their publication that happened after ours. And as you can see, this looks pretty similar to that. The trends are showing that amyloid is accruing at a very predictable rate, such that you can fit some kind of function to line it up, fit everybody on a curve. And our colleagues at Mayo Clinic over in Minnesota show the same kind of thing. They can fit all of their individuals, all these amyloid lines, to a single curve. We've seen this in the Baltimore longitudinal study on aging. We got the Baltimore data, Dr. Betthauser got the Baltimore data and did this with their data, fitting their amyloid scans to a single curve. And also in a big study called the ADME study, it's a big multi-site Alzheimer's imaging study all over the country, we get the same kind of thing. We can take this confused looking graphic on the left and fit each of these individuals to a single curve. And that means that this finding is replicable. We know that this is sturdy, this is going to hold up, and that's a big deal. This kind of thing will get to the clinic one day.
I know you're not going to say it, so I'm going to say it.
All right.
RAP was the first to do it. Science is a team sport, right? We work together, but it is nice to have something, a breakthrough where you're the first to do it. And Dr. Johnson, Dr. Betthauser, the whole team, Dr. Kosek, she's here, they were the ones. And so and others have learned from them, and they're using these sort of techniques.
We've seen that with this kind of technique, we can get an age of onset. And this is just plotting that estimated age of onset across all of our participants, both in RAP and in the Alzheimer's Research Center, which we partner with on a daily basis. And we have here, the dots are colored by a person's current diagnosis, not their diagnosis when they first had their amyloid scan, but their current diagnosis. The people in red are people with mild impairment or dementia, and you see that largely they began to have amyloid earlier in their life, in their 40s and 50s or early 60s. The average age when amyloid sets in and becomes detectable is about 62. But nobody here is average. You can see that those lines are all over the place, and it's best if we can take each person as they come. Think about them in terms of their amyloid onset, the duration of their amyloid disease, and go from there. And that's the value of this kind of study. Let me show you another example of how we can use this. We've plotted people here. Now the line represents their clinical symptoms. And this is a questionnaire that your study partners fill out. And we've had, this is a standard questionnaire that we've done all across our center. And the lines here represent progression on that standard questionnaire. It's called the Clinical Dementia Rating. And most people in RAP are very much around 0 on this questionnaire. But some people start to show elevations around 0.5 to 1 to 1.5 and so on. And when you get to the point of 4.5, you cross the threshold to mild dementia on this particular instrument. So what we see here are people that have really spanned the gamut of clinical symptoms. On the horizontal axis is how long a person has had amyloid in their brain. And we plotted the average again. I know none of you are average, but we plotted the average just so you can see what that looks like, and that's the blue line with the gray band around it. And on average, it takes people about 24 years from amyloid onset to reach the level of symptoms that we would call it mild dementia. 24 years, that's an incredible span of time. You see some people that are ahead of the curve that have reached symptoms sooner. These people tend to have more vascular disease in their brain, and they may be older, signifying or suggesting to us that there may be other diseases happening as well in their lives. There's also a group of people below that line, to the right of that blue line, who don't yet have symptoms, or their symptoms are much delayed relative to the others. These are resilient people or resistant people to this disease, and we really want to know what makes those people tick. Why are they showing resistance? And that's really the value of this project. And I want to thank all of those study partners who filled out that survey. And I know it can be annoying to get to fill this out on the study participant, and we'll call you and try to hound you for those questionnaires. We want to thank you for doing that. This is the value of that kind of questionnaire. Let's try to piece this together with a single participant. This is a case study. This is an individual who first joined the study in their late 50s, and you see their first cognitive testing right around age, 58, 59, and that's at that first red dot way at the top. And I also want to point out, well, and anyway, with these red dots, you can see their trend over time. And by their 5th study visit, they were down around the 7th percentile or so. But I want you to see that if this person was declining at a pace of normal aging, that red dot would actually be parallel to one of those dotted lines. So they're actually crossing lots of cognitive, what we call centiles or percentiles. They went from the 50th, they're well above the 50th percentile, down to the 7th percentile, to the point where they have clinical symptoms, and that's concerning to us, and we've identified that for them. But a point here to make is that we are comparing you to you, and that's the value of a longitudinal study. We don't use a lot of norms or population-based expectations, because those can be biased. We compare you to you, and that's the value of what I'm trying to demonstrate with this slide. The other thing down in bottom are these blue annotations. We have a PIB plus, which means this is when the person became they first became evident to be amyloid positive at that point. And that was, in their mid-40s roughly, early to mid-40s. And their plasma marker of amyloid and tau, those became positive, we estimate, in their 50s. And we'll talk to you more about plasma or the blood markers of this disease. So that's just putting a little bit of this together about how we can make RAP more personalized, not just for you, but hopefully this kind of technology will translate to the clinic in the near future. All right, well, let's talk about some of the other projects that have happened in the study. Do you want to go over those, Nate?
Yes. I just want to say that I was just at a national conference where the whole field was discussing how do we have better cognition outcomes and how do we test for the population, because there's such limitations in the tests that we take. How we normalize that is based on small numbers in certain parts of the country. And I introduced the fact that a RAP is doing this to themselves. These are participants who come in longitudinally and they're personalizing it. And that was sort of this, wow, okay, well, that's an interesting technique. Because when we test compared to you, we can kind of control for some of those limitations of just an inherent test. And that's a really powerful thing. Everyone was was really surprised and shocked and impressed that RAP was able to do it. And then I thought maybe I said too much, and so I said more. Sometimes I hear these things Dr. Johnson talks about, and I have to wait, oh, is that published? No, am I not allowed to talk about that? This is what RAP is known for, and this is why the testing matters so much, because of these very specific techniques that can be done, which certainly are going to translate to other studies, and hopefully then eventually in the clinic.
And Dr. Janitis is here, and she's working on a publication with this work. Thank you.
I hope I didn't do anything wrong. But I do want to highlight some of the great studies that have come to RAP and work with RAP. And really one of the things I wanted to mention is that RAP is really at the forefront of the importance of addressing health disparities, racial disparities in Alzheimer's disease research. I have a picture here of Gina Green Harris, who is based in the Milwaukee office of the Wisconsin Alzheimer's Institute. And when RAP first started, she got involved a few years later with this idea of how can we engage in the community? How can we reach out and see what matters to our participants in the community in which we're recruiting? And how can we keep this bi-directional relationship? And through the works of Gina Green Harris and Dr. Johnson and the colleagues at RAP, they really were able to improve the rates of African American participants from 0.8% to 8%. And that was really through this idea of transparency and honesty, these information sessions, and listening to what matters to the participants in the community. And that message is really carried forward. And one of our other colleagues, Dr. Carrie Gleason, has a grant called the African Americans Fighting Alzheimer's in Midlife, AA FAME. And she just got her renewal, so she has another 5 years to do this. And really, again, highlighting the importance of giving back. So listening to what matters and helping what participants feel matters impact the science itself, but then giving back through information. And that's going to be a common theme in the next few slides. But that has really led to, I believe, a closer connection, also the creation of advisory boards that we make sure we're doing things in the best way possible and in a way that impacts our participants. Through her work in the AA FAME, you can see here that in Madison there were 2 African Americans in RAP prior to 2017, and that number has gone up dramatically since then. But you can see that there are over 446 cognitive tests, and that has led to many MRI scans and PET scans and lumbar punctures and repeated visits. And it is really important for us to make sure that what we're showing in the science is applicable to every person, and making sure that that representation is seen not only in these observational, longitudinal studies, but then in the clinical trials, like the ones that I mentioned earlier.
So we are at 450 now, and we're on our way towards 500 African Americans between RAP and the Alzheimer's Research Center that we partner with. And that's, with those kinds of participation numbers, we can really make some discoveries on the things that affect that community.
And AA FAME participants are very active in other studies, some of the ones that I'll be mentioning next. And that's led to many publications. This is looking at vascular risk factors and Alzheimer's risk factors in blood flow in both groups of participants. And so, speaking of blood flow, we know that cardiovascular health is really important. This is why when we talk to one of our nurse practitioners, they're going, and we have one here today. Kara, can you raise your hand? She doesn't want to. Kara, raise your hand. She's a wonderful nurse practitioner who's going to talk to you about a healthy lifestyle, because we know that these things are important. We're figuring out how exactly this works, but Dr. Ozioma Okonkwo has some great studies looking at cerebral blood flow and vessel stiffness, and really showing that in this figure, I'm going to use the bottom right one, he has broken levels of fitness between low, middle, and high, and then looking at brain blood flow. And as you can see, the more cardiovascular, cardiorespiratory fitness you have, the better your brain blood flow. We know that's an important factor. He's also showing in the upper right figure that same thing. If the more fitness you have, the less the stiffness of your blood vessels. And that too is an important factor, because ultimately we're looking for ways to protect brains, and he's showing that high cardiovascular, cardiorespiratory fitness is going to lead to better blood flow, better, less stiffness, and ideally more resilience to changes that are happening in the brain. He's also looking at fitness when it comes to metabolism of sugar in your brain. Your brain does use sugar quite easily. And so in this regard, in the middle figure, the one that has the blue bar and the green bar, we have those that are active or inactive. So the active group is the green one. And on this vertical line is insulin resistance. We don't want insulin resistance. This is sort of a precursor to eventual diabetes. You want your brain cells to use sugar well through insulin. And so those that are active have less insulin resistance. And that's an important factor, not only for your whole body, but also for your brain. And then in the middle bottom one, with the different shades of green, he's broken, again, fitness down into low, middle, and high. And then he's looking, in this particular one, at your brain's ability to metabolize sugar. And the easier it is to metabolize sugar, it sort of reflects the healthier your brain cells are. And so the more fit you are, the better your metabolism is. The last thing I'm going to say, because I really like this figure, is the bottom right one. Those are PET scans. They're not the ones we've been talking about, amyloid and tau. It's a glucose metabolism PET scan. And so the more your brain is metabolizing sugar compared to its baseline, the greater the color. So in this regard, red is a good thing. And so what he is showing are those that improved in their fitness over his intervention, the more the improved health of the brain cells, in the front of the brain in particular, which is why you see that red color. So he's really showing that something I think we all feel is important, which is exercise and cardiorespiratory fitness. He's showing at the basic level and at the health level that these things really are important and will affect how our brain is functioning, and ideally how our brain can push back on diseases that may be occurring. But it isn't just cardiovascular fitness and blood flow. We know that where people live can impact their health too. And so Dr. Amy Kind and Dr. Barb Benlin have really taken great interest and effort in creating what's called the Neighborhood Atlas, which is a free open source tool. So if you were to Google this, you would be able to insert your zip code, and then a picture will come up showing levels of area disadvantage or area deprivation. It's a very validated tool using multiple factors, but ultimately coming down to access and resources and quality of the environment that a person is living in. And she's been able, Dr. Kind in particular, to show relationships between where people live and some of these biomarkers of Alzheimer's or cognitive health or physical health. And this tool is actually being used at the federal level, and it's helping shape healthcare policy and federal policy. And it all started here in UW. One of the factors when it comes to your environment is stress. And so Dr. Megan Zulsor is one of our great researchers looking at how, as she calls it, how stress gets under your skin. And we know that certain things, certain life events can be very stressful, but there's also chronic daily stress. And these are two different things, but they're looking at how does that impact your biology. In particular, your cortisol level. Having too high of cortisol for prolonged periods of time is not how our body was supposed to exist. And what is the impact of that? And certainly one of the answers is that it has a negative health impact, a negative brain impact, but she's looking at that factor. And so she's looking at the outcomes of brain health, of the biomarkers of Alzheimer's disease, and doing that through, I believe it's a cheek swab, looking at your cortisol levels. And it's pretty remarkable, but it really speaks to the importance of us addressing our mental health and certainly our stress. On the flip side of that is social support, so a positive thing. And she's looking at how the sense of community or support can impact a person's well-being and therefore perhaps how they are doing on thinking tests. And so the upper left figure here, you have sort of degrees of social support, so more support as you go left to right. And then on the vertical line is one particular memory test. And as you can see in both lines, the blue one representing elevated amyloid, the red non-elevated, the more support this group of participants had, the better they did on testing. Now that doesn't mean one's causing the other, but we see this relationship. And what I think is nice is it also doesn't matter if you have amyloid or not. So This is this idea of resilience. And so this is clearly an important factor that needs to be investigated more closely. And you can see it on the bottom right as well. Social support is on that horizontal line. And on the vertical line is a times test for complex problem solving. So in this regard, you want it to be low because it means you're quickly solving something. And so you can see with more support, individuals were faster on this thinking test. And again, that's actually the front of the brain too. So the one that I was showing you with Dr. O'Connell. So we see that there are these important factors. We do need to understand them better. But these things come from the questionnaires that you fill out and the information you give during your visit. So we do utilize that information when we try to understand other factors that affect your brain. This is a very popular and exciting one. And this is about stool and poop and looking at the gut microbiome in particular. Alfred is in the back. Alfred, if you could raise your hand, thank you. He's a part of the MARS study looking at, well, how does our gut microbiome affect our thinking, our well-being, certainly some of the biomarkers of Alzheimer's disease. And maybe there are people in the room who have donated and given their stool. It's a different type of sample collection. But we appreciate it because we are seeing there are these relationships. Again, not one causing the other, but there is a relationship between the diversity of someone's microbes that we measure in the stool and some of these biomarkers of Alzheimer's disease. This is a very rapidly progressing field. We're not the only site, although Dr. Feynman was certainly one of the first to really go forward in this field. But it's really an area of interest because it is, we know that our gut is our second brain. There's a lot of relationships in our nervous system between our brain and our gut. And so what are things that we can do? I think for those of us who like food, this is another one of those factors that matters. The foods that we eat influence our gut microbiome. And so how can we potentially alter that? And one of those thoughts could be probiotics, for instance, or certainly certain types of diets. But these are the things that she's looking at. She is continuing to collect samples. If people are interested, please talk to Alfred in the back. He can explain to you the process of collecting your stool and sending it in. I won't talk about it here. Everyone. And the last one I'm going to talk about is genetics. I mentioned to you earlier that the ApoE risk factor, and that is an important risk factor, but there are other genetics that matter. And so Dr. Corinne Engelman and her team really do look at the whole genome to see what could be protective genes. And actually we have another table in the back. Can you raise your hand for me? Cloco table, Team Cloco, and that's one of those potentially protective genes. And but then there are others that might increase risk. We know that ApoE is the greatest risk factor. Probably accounts for about 95% of genetic risk for developing not the early onset Alzheimer's, but our more traditional late onset. But depending on the other 5%, someone might develop symptoms sooner than others. And I think one of the fascinating things that Dr. Engelman talks about is that we still are able to impact our genes through a process of epigenetics. So the things that we experience in early, middle, or late life can impact how our genes are expressed. And so she's looking at that component as well. I'll end by just saying that there is, you might see in the news, polygenic risk score. It's multiple genes, putting it into, as Dr. Johnson calls it, a function. It's a formula, it's an algorithm, right? And being able to see that can help us predict who's at higher risk than others. And it's a very technical, and if she were here, she'd be able to explain it better than me. But we are looking at genes as one of those important factors in understanding, but also potentially quantifiable to some degree. I think this is one of our really exciting things that we wanted to share with you, and that is blood and blood-based biomarkers. So will there be a blood test? And the answer is certainly yes. We know that many studies have shown that these changes of Alzheimer's disease can be detected in a blood sample. It's not perfect yet, it certainly looks good, but there are things that we still have to understand. But in the end, blood is going to improve our access. It's going to increase accessibility to the community, to participants, and it's much easier to do. It's easier to get a blood test than it is to have someone come in to UW to do an MRI scan, PET scan, and lumbar puncture. And so we can increase the number of people engaged in science, which will ideally mean that we can reduce the time it takes to do science as well as clinical trials or monitor the changes when people have therapies. And so we still need to do our thinking tests and our PET scans and lumbar punctures to compare blood to those, but the direction is certainly one of getting blood. Some questions that the field is still trying and will answer going forward: how much amyloid and tau protein in the blood is considered abnormal. We still have those thresholds, although RAP is leading the way in that. What do we mean by abnormal? Who's determining abnormal and that threshold? How does it compare to the lumbar puncture, the spinal fluid, and the PET scan? Can it do more than just detect the presence of the protein? Can we help it utilize it to help predict change? And then is it generalizable to the entire population of people? And so in this figure here, on the horizontal line is the blood test. And as you go left to right, that blood test is picking more of that protein of Alzheimer's. And then on the vertical axis is the PET scan. And as you go from bottom up, the PET scan is detecting more of that amyloid protein. So you can see in the upper right where it says TP, or true positive, this is showing really strong, a really strong concordance or relationship between picking up the protein in the blood and having the protein in the amyloid scan. So these are very accurate when you look at that in particular. And then in the bottom left, where we have the T and an N for true negative, a low blood test score does look like it's very reflective of not detecting amyloid in the PET scan. So that's a very good finding and shows the accuracy of the blood test. And there's one on the, if you look at the numbers on the left side, there's one that says p-tau217. That's just the type of protein that we're looking at. But you can see 94% concordance or accuracy. So these blood tests are looking better and better. And we're able to do that. We're able to talk about that in the scientific community because participants come in to give blood and do the PET scan, or blood and the spinal test. But then what can we do with it other than identifying the protein? And in this regard, we're seeing that this blood test may be able to help us predict future change. And so on the horizontal line is age, so you're getting older as you go left to right, and then on the vertical line is your thinking ability. So it's changing, it's declining as you go top down. And in this regard, I want you to pay attention to the red line. Those are individuals who have a high amount of the protein in their blood. And you see over time on this thinking test that they're declining at a faster rate than those in the blue line who either have none of this protein or just a low level. So we are seeing this relationship. It's not something we're ready to put into a formula and have a prediction score for people, but it certainly shows that blood tests might be more helpful than just one thing. And we're looking at this in our diverse samples. Our African American participants have donated and consented to having their blood tested and then look at it compared to their thinking tests. And in this regard, these were 179 individuals who were cognitively healthy at their beginning, and then we were able to follow them over time. And we showed in the left figure is one of those complex thinking tests. And so again, you're getting older as you go left to right, and the red line reflects those with a high amount of that protein in the brain, or in the blood, in the blood. And you can see it's higher as you get older. And that just means it's taking you longer to complete that test. So having a high amount of protein in the blood seems to have a relationship with being slower in solving some of these problems on the thinking test. And on the right side is learning the number of words that we give you. And so the group in red, again, those are the ones with the high amounts of the protein. They're learning less over time. And individuals did not have impairments on testing. We just see this trend because the testing is so difficult and precise. So these types of studies have really led to a lot of publications that have come from RAP. And I believe in your folder you'll have a QR code that if you're interested, you can see some of the others. But RAP is really at the front of defining preclinical, defining blood-based biomarkers and how they can be used in the future. All right, I'll turn it over to Dr. Johnson.
Thank you, Dr. Chin. What's next? We told you we've got 5 years of renewed funding. What are we going to do that's the same? What are we going to do that might be different? We are continuing to add to the study. In particular, we are expanding our African American side of the cohort. We want to make sure we have sufficient representation so that our discoveries are generalizable to this community and other communities, and we're partnering with other communities as well. We are doing MRIs as usual, but we're expanding to Milwaukee for this. We're already in Milwaukee, and we're potentially expanding into La Crosse as well. We'll be adding ultrasound to the study, and the ultrasound is in the neck to look at the arteries that supply the brain, and we'll talk about that in a few minutes. But we are continuing to do most of our study procedures just as we have been doing them. The cognitive testing is our bread and butter, as Dr. Chin mentioned. And our blood draw is so important. It's becoming more important than ever. By the way, there's blood on many of you going back to when you started the study, in some cases back to the early 2000s. And that is now such a treasure trove to understand the beginnings of this disease based on the molecules of amyloid and tau that we can detect in that blood. Lumbar puncture and PET scans continue to be how we'll bridge the science in our field and the clinical care to blood. So we need pioneers like you to do both the blood draw and either a lumbar puncture or a PET scan, or both. If you're willing to do that with us, we appreciate those who've done it. We want to continue to talk to you about that so that we can get more people in those studies, as they are they're so critical. They speak so loudly with regard to the science, the discovery, the knowledge that will come from it. We know that Alzheimer's is the only thing that's happening as we age. Vascular disease is perhaps just as common, if not more common. It's not a proteinopathy, it's something that's happening in our blood vessels. People who have both of them, both vascular disease and Alzheimer's changes, we think they're changing faster. I showed you an example of that, and this is another example that Dr. Kosick recently put together. People who have both of these things in their brain are changing faster. And this is in RAP subjects who are largely pre-symptomatic. Even then, we're detecting a magnified effect on cognitive change if you add vascular disease with Alzheimer's disease, basically. Well, let's talk more about how we can measure vascular disease. This is done with MRI. You see that picture there on the right. That gray one is an MRI, and that's so important for understanding vascular disease. But we know not everybody can do an MRI. It's not always practical. So that's why we're developing this ultrasound methodology, and let's let Dr. Chin talk about that.
Did I see Dr. Rivera?
Oh yeah, there he is.
Can you get your hand up again for me? Dr. Rivera is one of our great scientists looking at blood flow in the brain, and he's doing it. He's created a technique, or using a technique that really is pushing the field forward. And so MRIs have some limitations to it, and we're really trying to expand what we can do with it. And I just wanted to pay special attention to that. Thank you, Dr. Rivera. Another way of looking at blood flow is the carotid artery that feeds the brain, and having an ultrasound. And so you can see a picture here of an ultrasound probe that goes onto your neck and looks at structurally your blood vessel, the carotid artery. And then the picture here, where you see intima media adventitia, that's the wall of your artery, the blood vessel. And so Dr. Jim Stein, who's the head of the preventative cardiology group at UW, was excited to join RAP in doing this study so that we can really get a better sense of the technique he's using and add another data point of understanding blood flow and how it might impact biomarkers, thinking ability, and so forth. And with that probe, you can see the thickness of the blood vessel. You can see the blood flow, which would be the white part on the top of that figure. You can see potentially if there's narrowing or if there's even a plaque there. And so he's able to really calculate a lot of different data points just from simply putting a probe on. And I'm going to skip the figure of that and just say that this is really safe. And these are the same types of probes that are put on ultrasounds when women are pregnant and being monitored. So there's not any real exposure risk. You can look at a lot of different things, as I've mentioned. And it actually can be helpful in predicting future events. And that's what Dr. Stein has been a part of prior to this. And so he's now going to bring that skill and expertise into the thinking world with us. We're excited to have him, and this will happen during visits for those that are able to do it. In addition to vascular disease, there's Lewy body disease. Probably some of you have heard of that condition, similar to Parkinson's disease. And Lewy body disease actually is the second most common neurodegenerative process of protein accumulation in the brain. And it can coexist with Alzheimer's disease and lead to accelerated thinking change. And actually, on a personal note, my father was diagnosed with Alzheimer's at 61, and he passed away at 66, which was a pretty fast course. And he was part of the study. He was very proud of that, although he didn't like the testing part. And when he passed away, he donated his brain. And sure enough, there was Alzheimer's disease, but there was also Lewy body disease. And as a family, we didn't know that. And it really provided information that was meaningful to us as to why did this happen so quickly. And really, we can only do that through an autopsy, which speaks to why if you have questions, I really encourage you to talk to Jay. My father really liked Jay. But also that these things, we don't know everything that's happening in the brain yet. And so through the autopsy we can do that. But also, now that we are looking at it more, now that, as Dr. Johnson said, 60% of people are going to have multiple things, well, knowing that is important because we're going to spend more time looking for other things too. And so that speaks to why we're going to have Kara examine you every time you come, because we're going to look for changes that might reflect something other than Alzheimer's disease. That's why the questionnaires matter, because some of those factors may be identified very early. And then lastly, there are two things that RAP is looking at and talking about implementing in the study. One is a smell test. Some of you might have heard of that. So it's a way of detecting whether someone has lost their sense of smell or to what degree, as well as looking at the spinal fluid that's collected and looking for the protein that forms and actually is what they use to define Lewy body and Parkinson's disease. It's called alpha-synuclein. And if anyone saw in the news the Michael J. Fox, the big breakthrough, Dr. Johnson's in communication with this group to see how can we potentially collaborate. And so people come to RAP because they know about the participants and the work that's done, and they want to expand our understanding of thinking ability. And along those lines, then what can we do as an organization, as a group, to really empower our participants, but also make sure that we're keeping in mind the ultimate outcome, which is improving people's health and maintaining ability. So I'm going to start with number 3. This is what we're currently doing. Anytime you have a clinical blood test with us, we want to share those results with you to make sure that if anything comes up that we talk to you. And again, direct you to Kara. She's wonderful at evaluating those things along with our other nurse practitioner team. In the middle, you'll see abnormal thinking tests. This is where we might call and talk about potentially you've seen some changes and we might want to connect you to a memory clinic or your primary care provider. We're now talking about MRI scans and ultrasounds, being able to share some of the key facts back with you from those. And then lastly, number 1, those biomarkers of Alzheimer's, so an amyloid PET scan. And that's once we as a field and as a center feel that a result is valid and meaningful to people, we want to be able to share that back with you. We need to have the infrastructure to do that, as well as the post-sharing process to make sure that we're there to answer questions and provide resources. But that is a value of RAP and something that we're going to continue to do and expand in the near future. And Dr. Lindsay Clark, one of our colleagues, has really led this effort in disclosing amyloid results. In the beginning, with a small number of people, it was to make sure it was feasible, that it was safe, and we really wanted to know what needs to come after sharing that result. And in the back is Sarah Albertson Frost. Sarah, please raise your hand. Thank you. She is leading the effort in sharing these results. So if you have questions about that, what does this mean for the future, please direct those questions to Sarah. But what we have found is that it is meaningful to people. And so we want to continue to understand and improve the process. We want to make sure that the services after learning your result are there for you and that we can utilize them. And at some point, we want to share more information. And so that's where more studies are going to be needed for that, but certainly RAP participants will be invited to join those. Along the lines of sharing is our new brain health report, which should go out in mail starting in the summer. This was really spearheaded by Dr. Johnson, who wants to give more information back and utilize the information you've shared with us. And so in this table here, and you can't see the details of it, but a lot of this is based on your lifestyle habits that you've shared with us. The foods, the sleep, the exercise, the things that we like to talk to you about too for your cardiorespiratory and glucose health. And so we will be able to input what you've shared with us and then compare that to sort of what the national guidelines are. And we can tell you the areas you're doing great and perhaps identify areas where maybe you could improve on, or if you wanted tips on ways that you might be able to do more. And so that's the brain health report, but along with that is going to be a picture of your brain, your actual brain. And so we wouldn't be using this clinically in our clinic, but this is going to be unique to you so that you can see what we are seeing when we look at your brain scans. And with that, I think I'm going to turn it over to Dr. Chin.
Thank you, Dr. Chin. I hope you've seen the impact that you've made on the study and on our field and the knowledge that we have about Alzheimer's disease. It's incredible, 10 years ago, to think that we would be able to talk about Alzheimer's in terms of an age of onset and a duration, and to talk about pre-symptomatic change like we've been able to do. We feel really confident in our methods, and that's why we're now able to disclose more of them to you. And we want to make the study be something that will benefit you as well as the communities that we come from. RAP is part of the Alzheimer's Institute. This is our academic home, and I just wanted to acknowledge the incredible support we get from WAI, or Wisconsin Alzheimer's Institute. WAI does a lot more than just RAP. They have a focus on health equity, on education of communities, as well as the healthcare system. They're really involved in educating physicians on how to identify this disease, make more accurate diagnoses, and have better clinical care. And our website is there, wai.wisc.edu, and it's headed by Dr. Carlson, whose picture is there in the middle of that slide. And I also wanted to point out something that's in your packet on becoming a memorable friend. This is from the Initiative to End Alzheimer's Disease, which is our philanthropy arm of our Alzheimer's program. The Initiative to End Alzheimer's Disease is here to answer questions and assist you if you have any desire to give in other ways besides being in a research study. We're not asking you to give more than you already are. Being a research volunteer is an incredible gift. But some people have expressed a desire to give more, including RAP or the Alzheimer's program in their estate planning or their wills and so forth. And so those kinds of things is something that UW is good at doing. So just wanted you to be aware of that. But the most important thing we wanted to do is thank you, our participants, your family, your friends, for supporting you in your journey and our journey together as we discover what is going on with Alzheimer's disease, and as we discover new ways to prevent and